Okay. Welcome everyone to another Kempner seminar series. So it is my great pleasure today to introduce Ila Fiete. Ila is a professor of brain and cognitive sciences, a member of the McGovern Institute, and the director of the Lisa Yang. It's a computational neuroscience center at MIT. I hope I covered all your titles. Ila is a leader in computational neuroscience and contributed to the field in many ways. Today I tried to go over her publication list and try to make my own summary of this. It turned out to be an impossible task. So I'll just say she's contributed a lot. And consequently, she's the recipient of many awards. And very recently, she was recognized by the Society for Neuroscience with the Schwartz Medal in Computational and Theoretical Neuroscience, which is the Nobel Prize of our field. So, this work particularly cited her work on modeling grid cells in cortex through pattern formation mechanisms in attractor networks. I really like the fact that they actually cited Turing pattern formation in that particular citation, which is great. That speaks to her PhD in Harvard physics also, as well, so she's a graduate of us. And then, of course, the work that follow up on that idea. So Ila's work has already shaped significant in neuroscience and will continue to do so. And I believe this is a intellectual continuity of that work. So I'm curious to hear what's going to come up. Thank you. Welcome again.
Thank you so much, Cengiz. That was exceptionally sweet and kind introduction. I hope I can live up to even a tenth of that in the talk today. So I'm very pleased to be here. It's wonderful to be back. I was here when Kempner just started, I believe, and it's amazing to see the community that's come together here and to see all the incredible Kempner fellows and the faculty mingling and talking to each other. It just seems like a very vibrant place. So congratulations, Bernardo and Tusham. Great. So I wanted to tell you about, as Cengiz said, it's very much a continuation of some work that we've been thinking about in the lab for a number of years. You know, when grid cells were first discovered, I was a postdoc, and I was alone as a postdoc because I was in the theoretical institute called the Kali Institute for Theoretical Physics in Santa Barbara, and there were no other neuroscientists there, and I was feeling quite lonely. And then I, read about grid cells, and it was crazy. It was like, gift for a physicist, and so I pretty much almost fell out of my chair, and then thought about, how do these arise, and then what are they good for? And that pretty much, I think, made up the arc of the next several years of my research work. And recently, I think there's been enough progress in the field, both among the experimentalists and the modelers of grid cells, that it felt like maybe one could feel bold enough to make the next step and start thinking about the areas that grid cells connect to, specifically hippocampal neurons, and try to understand them a bit better. And so that's my goal, is to try to tell you about what we've tried to understand in that space. First, most important is that this work was done by Sue Sharma, who was a postdoc, who was a grad student in my group, and by Sarthak Chandra, who's a postdoc in my group, and they're both super fantastic. This is basically their thing. So there's sort of two ways that I can motivate the questions about hippocampus that I want to address in the talk, and one comes more from a computational perspective, from the perspective of memory models, and like we just recently heard about Hopfield's Nobel Prize, which was fantastic for our field as computational neuroscientists. But the question is, what is a good model of biological memory? Okay, and we kind of, I guess, I would argue we lack good biological memory models. So the Hopfield network has been incredibly influential in sort of our conceptual understanding of how a collection of neurons can together interact and construct, long-lived states and recall memories, and error correct and pattern complete, through interactions and through their dynamics. But at the same time, it has some pathological properties. So in Hopfield networks, you can add in, if it's a network of order n neurons, you can add in of order n, like information dense patterns into the circuit, and it can memorize all of them. In fact, it memorizes them extremely well, like to the point where it would be like memorizing white noise static, on a screen if you see that as your pattern. But then once you start pushing in more patterns beyond this capacity of order n patterns, then what happens is this breakdown, and the network is not capable of storing any more patterns. In fact, if you shove in more patterns, it also forgets everything that came before. So this is like worse than what you call in machine learning, what's called catastrophic forgetting is not nearly as catastrophic as this. This is actually total and complete breakdown of all the past information that's stored. So I'll just call that the memory cliff. And there have been, of course, in the many decades since the first proposal of Hopfield networks, there have been many alternative memory models or different flavors of these Hopfield networks that have been proposed, like including sparse Hopfield networks or Hopfield networks with sparse inputs, different kinds of sparsity, and all of these models have a cliff. They just happen at different points, and they all touch this sort of so there's this sort of envelope over here, which I'll say is the theoretical bound of order n squared, where this is a network of n neurons with n squared synapses. So that's the total amount of information, the sort of bound on Hopfield networks, and how much information can be stored in them. But you can see that even with this fundamental bound, these models aren't really living along this envelope. They're all coming to this envelope and touching it at one point, and then they break down, right? So there's the cliff. There's even more modern iterations of Hopfield networks, and these have similar behavior. So the question is there like a memory model one could write down that sits closer to this envelope over a longer regime? So are there models where we can trade off, like as you add in more and more patterns, is there a sense in which one could stay close to this envelope by having less detail that's recallable per pattern, but more patterns stored? Okay, and so is there such a model that exhibits this kind of trade-off? And the second thing to note from the computational perspective is we also have not very many, models of episodic or sequence memory that have a capacity that scales well with the size of the network. Okay, so that's sort of the computational motivation of thinking about memory and hippocampus and computational models of the hippocampus. The other motivation for trying to understand hippocampus and memory is a biological one, which is that what we see in neuroscience is that, so here's the diagram, the famous Fellman-Van Essen diagram of the visual hierarchy, and it was made to illustrate the complexity of all the interactions of areas involved in visual processing. But at the very top of this hierarchy is actually are these two areas, ER and HC, and which stand for entorhinal cortex and hippocampus. And so it's funny, some visual neuroscientists usually ignore those areas and think that these are pasted on. They're not. The entorhinal cortex and hippocampus are at the very top of this visual hierarchy. In fact, they're at the top of all of the cortical hierarchies, because entorhinal cortex is association cortex and hippocampus gets inputs from entorhinal cortex. What's biologically sort of puzzling about the hippocampus is that from rodent studies, and now also various other mammalian neuroscience studies, we know that the hippocampus contains clearly spatial responding neurons that are firing as a function of 2D explored space. So this is an example of grid cell, and this is an example of a place cell. This is a trajectory of an animal running around in this square box, and all these red dots here are spikes from a single grid cell over here that form a nice triangular lattice, and here a single place cell, and it fires in the neighborhood of one location, right? And so clear spatial correlates, very specific seeming tuning for spatial coordinates, right? So it seems like a very special circuitry for spatial representation. But at the same time, we also know from human studies that the hippocampus is essential for autobiographical memory, episodic memory, the kinds of memory that you use to remember whom you spoke to and what you talked about, what you were eating at the time, and where that was. This is known to us from most famously from the patient HM studied by Scoville and Milner, who described a total inability of HM, the patient, to form new memories after he had surgical resection of part of his hippocampus and entorhinal cortex. And so, you know, although his semantic memory was intact, he was able to tell you, for example, that Paris was the capital of France, but he wasn't able to tell you after meeting you today that earlier he had met you. Okay, so on the spatial side, we actually have models of, you know, grid cell formation, maybe some spatial tuning in hippocampal neurons, but we kind of lack models of episodic and autobiographical memory. And so, you know, we can make a few different hypotheses, right? Given this biological, you know, this observation that hippocampus, you know, there coexist two different types of memory in hippocampus, the spatial memory and the episodic memory. You know, we can ask, you know, why they're co-localized. And so one hypothesis is that this circuit is optimized, is optimal for spatial memory, right? Spatial memory is super important, you know. Where was that tree where I found the fruit, right? And so, you know, that's kind of, you know, what our circuit is really, the primacy of the circuit is for spatial memory. And then episodic memory kind of, you know, rides on, right? It, you know, it's there as an augmentation. The alternative is that this circuit is, you know, really optimal for episodic memory. That's what it's for. And then it's just the spatial components. The reason why we see any spatial tuning in neurons in the circuit is because space is a useful index, right? It's a way to retrieve the episodic memory. It's reliable and it's stable, right? And so that's the idea. And that's why we see these like grid cell type representations, just a spatial index. Okay, and then there's a third hypothesis, and these are not, you know, mutually exclusive, but our third hypothesis here would be that, you know, actually the same very structured representations that you see that we ascribe as being spatial representations, for example, grid cell-like tuning, are in fact optimal for spatial and non-spatial episodic memory, even when there is no spatial information involved in the coding. And that's what I'm, you know, hoping to describe. Okay, so before jumping into the full model, I just want to spend just a couple of minutes going over the, you know, two critical properties of grid cells that will be important for these models. So the first one is the observation that's now being confirmed over, you know, basically ever since the discovery of grid cells, is that the grid cell circuit is first of all has a low dimensional set of states. So each, you know, set of grid cells that have similar spatial period are actually just forming a set of states in their population that lies on one two-dimensional torus, and that these states are invariant across conditions. So whether the animal is awake or asleep, these states are present. Okay, and the circuit only occupies these states. So even though it's a 10,000 neuron circuit, the states that the neurons in the circuit occupy are just on that surface of that one torus that's two-dimensional and nowhere else. And so various different studies, you know, tried to test whether that's true. And so this was looking across environments, familiar environments and novel environments, as well as rescaled environments where the spatial tuning of the grid cells was changing. But the finding was that the cell-cell relationships were preserved and consistent with all the states remaining on this low-dimensional manifold. The same thing is true when animals are put into a 1D environment, although their tuning curves look in one dimension, not periodic, and not like simple shifts of one another. Nevertheless, their responses are actually consistent with parallel slices through two dimensions and consistent with the states remaining on this same conserved 2D torus, even though these tuning curves look quite distinct. This is a study showing that across waking and sleep, if you look at the structure of cell-cell relationships and correlations between cells during running in green and during sleep in orange and purple, you see that the correlation structure is completely preserved in the grid cell circuit, whereas it's not preserved in run in green versus in sleep in purple and orange during, I'm sorry, green in waking and purple and orange during sleep. The correlation structure is different in the downstream hippocampus. And most recently, studies that were able to access hundreds of simultaneously recorded grid cells, simultaneous grid cells in the entorhinal cortex from the Moser lab showed that indeed much more directly by constructing these population states and embedding them in a visualizable way, we're able to confirm that the states lie on this 2D torus, and the states remain the same across a bunch of different environments and across waking and sleep. Okay, so this sort of invariance and low dimensionality of the circuit is now, I think, a very, it's kind of like it's sort of a cornerstone that we can rely on when building models that include these cells. Okay, so now the other property that I should mention that's going to be relevant is that the grid cells, that they're coding properties. So grid cells come in many flavors. So they have, which are many different periods. So they're adjacent neurons nearby each other in the cortex have similar periods. And then there are clusters of neurons as you move from the dorsal to the ventral end of this region of entorhinal cortex that have progressively bigger periods, but in discrete jumps. So it's a group of cells has one period, and then another period, and so on. So there's about a handful of distinct periods that grid cells exhibit. And so if we think about the grid cell code as, because it's periodic, it's representing position as just a phase relative to its periodic response, right? So it's just a 2D phase code for position. Okay, that's true for one population of grid cells with one common period. It's one 2D phase, so it's a circular variable, right? And or 2D circular variable. But now if we just think about coding one dimension in space, then a grid code, one set of neurons with the common period, is just one circular variable, which is just this 1D phase. So now we can talk about multiple different grid modules, which have different periods, and each of them is defining a circular phase, right? And so if we have n different modules, and actually in the brain, there are a handful of order 5 to 10 different grid modules, then they define 5 to 10 different circular variables. So if we just think about the code, so let's just think about this as the spatial position variable, 1D, okay? And we're moving, we're expanding the range of x going from 0 out to some distance over here. And this is actually two circular phases. This is the phase in this dimension of the torus, module 1. This is the phase of along the second module. So this is module 2's phase. Okay, and so what happens is as you scroll position starting from 0, maybe this point, then and position moves to the right, then the phase along each of the modules is changing. Okay, but because the periods are not identical, then by the time the first period the first module has wrapped around in phase, the other module hasn't quite wrapped around in phase yet, right? So basically you get, slightly incommensurate wrappings, and that then you can keep going. And that actually generically, for generic choices of lambda, defines a space filling curve on the torus. And so, in fact, the coding length that you can get of unique codes for positions scales with the number of different modules in the exponent. Okay, so what's interesting about this code is that, and this is also true even if you assume the code has, the line has finite thickness, so it's, it's some finite resolution with which you can read out the phase. Nevertheless, the coding line has this exponential capacity. So, so this is interesting because first of all, it has this, exponential capacity using linearly many modules and therefore linearly many neurons. Okay, but it's not just an exponential code with linearly many neurons. I mean, any sort of combinatorial code would be like exponential in capacity, like words of length 3 in the English language, right? That's, a lot of different combinations. But what's really interesting about this is it's combinatorial or exponential in capacity while maintaining this very structured algebraic coding property, which is that, as position changes, then the phases change in a predictable way. So in fact, so there's sort of this algebraic or vector relationships that are preserved, and the code word for position x plus the code word for position y is equal to the code word for position x plus y, which is a very important thing if what you're doing is representing position and then moving, and then as you move, the code word needs to update to then represent a consistent position variable. Right? So the combination of exponential coding and this algebraic property is quite interesting and special. Okay, so now with that background for grid cells, now we're in a position to talk about the bigger circuit and try to understand the hippocampus as a combined spatial and non-spatial memory model. Okay, so let me just show you the architecture of the full circuit and then we can unpack it. So here is our set of grid cells. Grid module 1, grid module 2, and grid module 3. These are each these attractor networks where the set of neurons here expresses just states which correspond to, a pattern and all translations of that pattern, a periodic pattern. And this is a second module, and then these do the same thing with a different period, and this one does the same thing again, right, with a different period. So these are our grid cell networks. Our grid cell networks can receive some velocity input to update their phases and shift the phase of the pattern, and that's kind of the existing models of grid cells. And now these grid cells project to hippocampus in a special way. So this projection here is random and fixed. The dashed line is fixed random connectivity. And the return projections here from hippocampus back to grid cells are learned once and then fixed. And they're learned just associatively for self-consistency. So the idea is that they're learned so that whatever grid state through random projection creates a certain hippocampal state, then the hippocampal state projects back and tries to reconstruct the same grid state. Okay, so it's just a self-consistency associatively learned set of weights. And finally, then these the hippocampus projects out to cortex, neocortex, with some associatively learned weights, and there's a return connection that's also learned associatively. Okay, so that's it. That's the structure of the whole circuit. These recurrent weights here enforce these patterns and enforce this low-dimensional dynamics in the grid cells. Okay, so in a nutshell, then you have these sort of low-dimensional grids. You've got a low-dimensional update mechanism. So these velocity inputs are just two-dimensional because they just drive shifts of this two-dimensional phase of each grid module. Okay, so the velocity input is assumed to be two-dimensional. And then we've got these rigid grid hippocampal connections and interactions with Hebbian-like updating of all the remaining weights. Okay, so this is the circuit model. And in fact, today we'll talk about a few variations in this basic circuit. So one is just this exact circuit, but without this velocity input. And the next one is going to be the circuit with the velocity input. And without the velocity input, this is a model of item memory. With the velocity input, it's a model of spatial memory. With a velocity input, which is a more generalized notion of velocity, where transitions are driven from the hippocampus, this would be our model of episodic and sequence memory. And then the same model can be used to understand a very interesting phenomenon called the memory palace technique, which hopefully I can get to at the end. Okay, so this model, I'll just for short refer to it as vector hash, which stands for vector hippocampal scaffolded heteroassociative memory. It's a mouthful, but I'll hopefully this will make sense, the name of it in a bit. Okay, so this model has two components. This first two areas, which are the grid hippocampal interactions, which I'll call the scaffold, and then the hippocampal neocortical interactions, which I'll call the heteroassociation part. Okay, and we'll unpack this. Okay, so now the whole question is, all right, I've defined a model, but what are its computational properties? So let's talk about first the computational properties of the scaffold. So what the scaffold does is high level this network, which is grid states randomly projecting to hippocampus with self-consistent projections back. My claim is that this network converts the exponentially many grid coding states into exponentially many robust fixed points of the whole circuit. Okay, because the states that are expressed by the grid cell, multi-module grid cells, those were not fixed points because each module operates independently of every other module. Okay, but by coupling to this hippocampus with these random projections, it creates exponentially many very well-behaved, nice fixed points. Okay, and that's what I want to show. So if we wire this up this way, first empirically, this is what can happen. So we've got some initial grid state that drives some hippocampal state through its random projections. Now if we add noise to this hippocampal state and then pass it back up, the state back up to the grid state, the grid state, the return projection to the grid state and the recurrent grid connections, do a bunch of cleanup, and then restore the state back to that hippocampal state. Okay, yeah.
And what's happening with the picture? This is all with the grid cells. What about the non-grids?
I'm sorry. Oh, so I'm only modeling grid cells. So we can think about, so right. So going back to this one, these are only grid cells in entorhinal cortex. And this is, you can think about these as direct neocortical projections into hippocampus, like for example, PFC, but also the non-grid entorhinal cells.
I think I was just wondering what are they doing in this?
In this picture, I've excluded them. I'm only showing you the scaffold network, right? So only these top two components, and we'll talk about the later part. Yeah. Please ask questions if they are asked. Okay, so all right. So what happens now with this scaffold network? Okay, is that it's able to restore hippocampal states that have noise added to them, and the question is, what is quantitatively the property of the circuit? So it turns out that the number of fixed points in this network as a function of the number of grid modules for sufficiently many hippocampal neurons, okay, it is scaling. So here you can see I can construct exponentially many fixed points with linearly many grid modules, okay? So that means, and modestly increasing number of hippocampal neurons. And in fact, if we look at the minimum number of hippocampal neurons to enable this number of points to be fixed points, that minimal number of hippocampal neurons scales only linearly with the number of grid modules, and therefore logarithmically with the number of fixed points. Yes?
I don't understand this plot. So you have number of grid modules, 8.
Yes.
So e to the 8, not 10 to the 12.
No, it's not e to the 8. It's exponential in the number of modules with like but with the but it's actually more than that, right? It's not just e to the 8. It's like with the big prefactor as well, because I mean there's many phases. So in fact, it's not linear just in the number of grid modules. I mean, it's not exponential in just the number of grid modules, but it's got a prefactor, which is how many different states are within each grid module.
But there is a big prefactor.
There's a big prefactor and there's a big exponential factor, but is that, yeah. So it's actually, so it's, okay, so it's not e to the n, okay, if n is the number of modules, it's lambda to the n, where lambda is the number of states per module, and n is then the number of modules. So that's what it is. Yeah. So if they're like, in each module, say you have a phase resolution where you can resolve 10 phases, then we have 5 modules, then it's going to be 10 to the 5. That's the, yeah.
Fine. 8. So I'm trying to understand.
Yes.
What's important? Number of modules are 4 or 6.
Yeah. That's right. So
Coming from something.
it's coming from 10, right? So we're saying that there's like, so this is a case where we've got, say, 11 phases per module. So then it would be 11 to the power 8 here. That would be the number. So instead of e to the 8, it's 11 to the 8. That hopefully clarifies.
So if there were 4 modules, something wrong with that?
I'm sorry?
4 modules.
Yeah, with 4 modules, you get 11 to the 4. So, that's okay. No, nothing's wrong with it.
There's no magic in the number of modules because there's a small number of modules.
Yeah, that's right. It's a small number, that's right. But I think what's important here is you get this very large number of fixed points, right? As a function of, as you scale the number of.
The main point is that the big increase in the number of hippocampal.
Yeah, that is the main point. That's right. You don't need a very large number of hippocampal neurons to get this exponentially many fixed points. Exactly. Yeah, that's right. So it's only linear in the number of modules and therefore logarithmic in the number of fixed points, right? That's all we need. And more even so that's the number of fixed points. And interestingly, all of these fixed points have identical size basins, and the basins are convex, and also the basins are large. Okay, and we can analytically prove this. But here, I'm at least showing you the numerical verification. So what you're seeing here is if we add this noise in the hippocampal state, okay, then this is the amplitude of the noise. So this is the magnitude of that perturbation that's added, divided by the norm of the non-noisy hippocampal state. Okay, and that's what we're showing here. And then looking at the probability that after adding that amount of noise, the circuit corrects the state back to the correct basin. And you can see that this is a high probability, even out to noise magnitudes that are of order 5 times larger than the magnitude of the hippocampal state itself. So these are large, and the different gray curves are actually the experiment done for different basins. And each basin, because of the symmetry of these grid states, all the basins end up being identical in size and convex. Okay, and here is to illustrate for you that what really matters is the relational structure between grid and hippocampal states that's caused by this fixed random embedding from this expansion from grid states to hippocampal states through this random projection. Because if we take the same set of grid states and the same set of hippocampal states, but then shuffle the relationship to each other, so which hippocampal state is assigned to which grid state, and then bidirectionally learn this these connections so that they can be well associated with one another as well as possible with associative learning, then the capacity of the system collapses. Okay, so it's the fact that these hippocampal states are derived through this fixed random projection from grid states that makes this possible. All right. So okay, so now this, okay, and none of this is, so this means that these basins are well structured, they're well separated from one another, but this is not yet a memory because the states that are being stored here as fixed points, as these stable fixed points, are not like user-defined states, right? These are the grid states themselves that have become, and these hippocampal projection states that have become the fixed points of this dynamics. It's not content that we choose to store in the network. So now this is the memory part. So the idea now is very simple, is memory, given any input, like a sensory state, it can be, yeah.
So here, what are the nature of the hippocampal states?
The hippocampal states, oh yeah, good question. So these are constructed by, you take the grid states, apply a fixed random matrix that projects out to this higher dimensional hippocampal state, and then those are just thresholding their responses. So there's a pointwise nonlinearity. Random projection,
the states are just.
Yes, the hippocampal states are just random projection of the grid states with the threshold.
They're not place.
They're not, well, yes, they need not be place cells in this case.
They're not place cells.
They're not, that's right, in this case. But in the spatial model, the same projection supports place cells if the grid states are grid cells, right? Because there's no spatial anything here, right? We're not talking about space at all, right? Like we're just talking about states. There's no space.
I'm not usual place there. There's something else. It's different neurons, or it's just,
it's just, right now we're just talking about states because we don't have space. Like I'm not talking about, like.
The grid cells have the same state as they have when there is space.
Yeah, but you can just think about it as a list of states, right? Like the grid cell states, we can just think of it as a list of states. We're not associating them to place right now, right? So we will come back to talk about space.
But
so
two-dimensional coding.
Yes, we will come back to that. But right now, yeah, there are states that, if you were to superpose them onto physical space, it would be grid-like. Yeah, it's a 2D code. That I agree. It's a 2D code, but yeah, no place. So then you're saying, yeah, what do these hippocampal states look like? In that same space, they actually do look like place cells. Yeah, come back to that. And if I don't, then we can discuss again at that point. Other questions? Yeah.
Train the upper left, the heavy and
the return weight. Yeah.
You actually have to navigate all of grid cell.
That's an excellent question. And in fact, we will, I think, talk about this. Oh, I don't talk about this. Yeah, that's a very important point. And I took this out, I think, for a time, but it's a very important point. So in fact, what's really interesting about the structure of the grid code is that you do not need to traverse all of these points. In fact, the number of points here is like the period with exponentiated to the number of modules. But in fact, the number of states you need to traverse to learn the return weights is just one module length. So it's just, it's linear. It's instead of exponential. So it's a very small subset of the total number of states as long as you traverse them in order. So you can't just visit these states in random order, if they traverse them in a sequence of like, like corresponding to a velocity, for example, in 2D space, then you only have to go through very few. So we call this strong generalization because you just visit very few states and then it learns the whole scaffold. Yeah, and it's all, again, because of the symmetries of the grid code. Great. Okay, so now the idea for now storing memories, user-defined memories, is very simple. It's just by associating or hooking these new sensory inputs that are provided from the external world onto one of the fixed points in the scaffold network through associative learning. So we call this heteroassociation because you're associating these inputs onto this set of fixed points that exists. Okay, so now if this is like the pattern that's coming in here, we get the pattern, activate one of the fixed points, and then with simple associative learning, hook this pattern onto that fixed point. If this is the memorized pattern, then you can take a noisy version of that pattern, drive the hippocampal state, pass the whole system through, hippocampal to grid to hippocampal and back, and you get a recovered pattern over here. That's a denoise version of the pattern. And in fact, you can do the same thing with much more corrupted versions of the pattern and then come back again to retrieve the pattern. What's also interesting here is that, so that, I'll just say that. And now the other thing here is you can ask, what is the nature of recall in this circuit? How good is your reconstruction of, information given noisy inputs? And so you can see, I think over here, that this pattern that's recovered is denoised relative to the noisy pattern, but it's not exactly the same as the memorized pattern, okay? And in fact, even if you feed the network a clean pattern, it will bring you to a slightly different pattern, okay? So it always brings you to the correct basin, but it brings you maybe not exactly to the state that was memorized. Right? Yeah.
So what is the capacity of the heteroassociative?
The capacity of the heteroassociative network is it's constrained by the size of the hippocampal layer, of course. Nothing is violated about the usual, exactly. So nothing is violated about the, fundamental capacity bounds in circuits, like, the Gardner results or the results from Abu Mustafa, those hold, right? And it's
no exponential.
I'm sorry?
No exponential.
There'll be an exponential in the number of patterns. Like we'll get the nice trade-off, and I'll come to that in the very next slide, which I'll try to show you. So total information is fixed, but then you can trade off number of patterns and the amount of detail you have per pattern. So let me just set that stage actually right here. So basically, if we look at number of hippocampal cells and look at the number of states that can be recalled, right, stored and then recalled faithfully, then as you increase the number of hippocampal cells, you can store an increasing number of patterns, okay, up to the number, and in fact, as the hippocampal size gets sufficiently big, you can basically almost put in as many patterns as the number of grid states, in the sense that the network correctly retrieves the hippocampal state associated with those patterns. It also correctly retrieves the grid state associated with that pattern out to exponentially many patterns. It also recovers the right basin in the sensory domain. But what it doesn't recover perfectly is the details, like pixel level details of the sensory input. And so, although the amount of information, of course, grows as you increase the hippocampal size, but this is not close to perfect recall. Okay, so so this is the result over here, and in fact, then we can look at the the capacity curve over here for this network for vector hash. So this is the mutual information that's recovered per input bit, okay, as a function of the number of stored patterns. So we can look at this in the log-log plot, this inset over here. So out to the size of the hippocampus, this network exactly reconstructs every input pattern, right? Every bit of every input pattern out to a number of patterns equaling the size of the hippocampus. But once the number of patterns grows beyond the size of the hippocampus, the amount of information recovered per input bit is dropping, okay? But the number of patterns is increasing, right? So in other words, this is a network that's staying close to that n squared information bound, which is the dashed line, okay, by trading off pattern recall richness for number of stored patterns. Okay, yeah, questions, I see, yeah. Go ahead.
Really elegant dynamics between the grid cells and the hippocampus to make those states into fixed points. I have a really dumb question. Why does the sensory input go into the hippocampus rather than through the grid cell? Like in my memory of the diagram, right, the sensory input gets processed.
Oh, yeah.
Is the input, hippocampus is the end stage. And so
that's a great question. I'm glad you asked. So I should clarify it. So this sensory area that we're talking about is, it's the non-grid cells of the entorhinal cortex.
Thank you.
And any direct neocortical projections into hippocampus. So it's like PFC and non-grid EC.
Okay.
That's what that is, right? And then the top layer is just only the grid cells, which is a small, 20% of the cells in EC are grid cells, and that's that. Yeah.
Is this assuming the patterns are like equally independent? Because like you could categorize them more efficiently and associatively, and then it might be able to store much more information.
Yeah.
So in this particular, I forget whether for this graph, I mean, we've done both. So we have correlated patterns and uncorrelated patterns. So this graph, I think on the left is just uncorrelated random inputs. And then, oh yeah, here it is. These are random memorized patterns. And then later I'll show you some results for also correlated patterns. Okay, and so by comparison, so once again, this is now a different depiction. This is mutual information per network synapse as a function of the number of memorized patterns times the bits per pattern per synapse, like the amount of information that was shoved into the network versus the amount that each synapse can recover of the information that that was put into it. And so a perfect system would, the mutual information recoverable per synapse is 1, right? Or it would be at least some finite constant. And this is what vector hash does, is it asymptotically it remains close to of order 1. I mean, it's less than a half, but it's somewhere there. Whereas various flavors of Hopfield network, have this memory cliff that you can see over here. So that's the result. So in other words, this network is able to do high capacity item memory, but without having a memory cliff and exhibiting a smooth trade-off between pattern number and detail. Okay, so what's interesting from, just to connect with some of the stuff that some of you think about, in machine learning, the question is, all right, we can think about this memory network, this network over here, as an autoencoder, right? Because we're passing in an input, putting it through the circuit, and then trying to reconstruct that state. And so, how well does an autoencoder work if everything was fully end-to-end gradient trainable to just reconstruct those patterns, right? That seems like a fair baseline. So what we can do then is, this is just a depiction of vector hash just unfolded, so that this is like the bottom-up pass, and this is the top-down pass over here. So again, the sensory inputs going to hippocampus, going to grid cells, back to sensory and hippocampus. And again, like the weights are completely fixed from hippocampus to grid and grid to hippocampus, and the recurrent weights within the grid layer are completely fixed. And the network that we compare it with is an autoencoder where each of these layers is exactly the same size as each of those layers. And now instead of this recurrence over here within, for memory to have the pattern completion, we just have an identity tail biting weight over here, so that the output here goes back into the input here, and so the network can iterate. But all the other weights here, all the gray weights, are trainable and learned through gradient learning. Okay, so when we do this, then this is what we find. So this is the capacity kind of plot for vector hash again, like you saw, number of memorized patterns, and then cosine similarity between the stored pattern and the recalled pattern. And you can see that vector hash again maintains, this. If we looked at this on a log-log scale, it would be again close to this dashed line, which is the bound. And here is the performance of the fully gradient trained autoencoder.
So this is the problem with architecture of the learning.
I'm sorry, Haim?
This is the problem in the autoencoder of the architecture or of the learning?
I think it must be a problem of the
design. Definitely below an inch I can write down.
Yeah.
Without the grid, which would give you perfect.
Yeah, below NH, you probably could. That's right. So
So it's not the right.
it's not, yeah. So you're saying that you're just saying that there's already a small performance drop relative to 1 over here, right? And so maybe it's the learning rule. It's in part. And I guess one could also answer that. That's a good point. One could, we could do that more carefully. There's also we could also add recurrence and have those recurrent weights fully trainable as well within the internal layer. But yeah, so that's a good point. We could try doing that. But you can see, I mean, it's not that far from 1, but your point is well taken. But you see, there is still going to be a crash at NH in this model. And here are some examples of what you see. So these are correlated patterns related to the question that was asked earlier. So here is the item that's stored, and then here is the recovery as a function of the number of other intervening patterns that were stored in the network. And so here you can see in vector hash that there's a graceful degradation of the quality of the recall for this pattern over here as more and more patterns are stored. And here, on the other hand, is the performance of the tail biting encoder, autoencoder on the same pattern with the same intermediate patterns. And you can see that after basically it does pretty well for a small number of patterns, and then pretty much it's basically not even trying to recover the pattern. It's just doing something completely different. And the same is true for another example. So this seems, I mean, I guess like one of those advantages, I think, of these biological constraints or these inductive biases here, this particular architecture, which is kind of shocking that you build an architecture where the internal representations are completely fixed to be grid representations, and then these fixed weights, and this network outperforms a fully trainable architecture, which is quite interesting. Okay. So, and this is also true, under the case of, with noisy memories, or if we take away the tail biting step, it's still true. So, okay, so why does all of this work? So conceptually, I think the idea here is that in a Hopfield network, when you train it to store, you give input patterns, then it's creating minima at the locations of those input patterns, but those input patterns dictate the locations of those minima. And so the minima can be, uneven, jagged, close to each other, not evenly spaced. Whereas over here, the network is, because of the symmetries of the grid code, it creates these like very well spaced patterns that make these nice convex basins, and now the content is simply stored by just hooking onto those basins. And so I like to think of this as like a clothesline, where the clothesline is this fixed thing, and then you can hang any item of clothing that you want out to dry. Okay, so that's kind of the intermediate point, and just conceptually what we needed is we basically factorize in memory. You've got sort of the content storage part, and then you've got the dynamics that does the auto-completion, the pattern completion, right, dynamics. And we've just factorized those problems. So there's one part that's doing the pattern completion dynamics, and then there's another part that's just doing the reconstruction of the content. And so this factorization is what makes this interesting. And the grid code, of course, provides the symmetries to make the scaffold high capacity. And we require these like random projections from grid to hippocampus. And the knee in this memory curve is determined by the size of the hippocampal network. All right, so now spatial memory. And so the same network now we can hook, we can now relate these states of the grid code and the hippocampal codes to the external world by taking movements in external space of an agent and using those velocity inputs to drive the grid states, right? And thereby we are mapping these internal states to now 2D space. And so when you do this, this network is now allowing, because it's building these associations between the velocity-driven internal grid states and the sensory-driven hippocampal states, it allows bidirectional inference where, given a seen landmark, driving the hippocampal state, it can reconstruct the grid state, so therefore infer position given a landmark. And on the other hand, given a position, which is a known grid state, but an unseen landmark, it can then reconstruct a hippocampal state and an inferred landmark, and thereby predict what landmark should be visible at that location. This model can also do zero-shot inference in novel environments. So if this is a novel, if there's a novel environment and the agent traverses a loop in this environment over here through this white path with these particular landmarks at each of these locations in the environment, then on a subsequent path that is a completely novel path that intersects those locations, this model through this pathway can then infer and predict what landmarks should be there, even when it has not taken that path before. And finally, if you now look for, like, we just run this agent through the space and probe the spatial representations, then the neurons in the grid cell area, of course, by construction, have grid cell tuning, and this randomly generated responses in hippocampus have a structure that looks like place cells. Yeah.
Why isn't it perfect in the new path? I mean, it seems like being at a location should recover the grid cell perfectly. We should recover the pattern.
Oh, these patterns? That's a good question. So just to make it a bit realistic, we just assume that this agent, this network had already memorized a bunch of other environments. And so it's kind of on the continuum of its memory curve where its reconstructions of the sensory states are not perfect. So that's why. But if, yeah, that's it. And I think Chaim had.
So if you have another sensor input where there is a landmark instead of the bird in the same position, there will be something else, a squirrel. Will it identify that it's the same location?
Well, there would be a mismatch between the internal grid state estimate of its position and the landmark-driven estimate of its position. And in fact, we're just trying to understand right now the dynamics of like which of those wins. And, then to use that as, but what's really interesting is that already the mismatch could give you a cue that maybe you should remap, right? You should just like, okay, there's something I don't understand about this system. So
both of them are the same. I mean, just observe landmark 1 and observe landmark 2. So there's nothing to be understood. I mean, I'm just saying there is no reason why there should be a systematic index of location of the landmark in this mechanism.
Okay, I thought you were asking a different question. So what this agent is doing is we started at some grid phase, right? It moves through this environment. Grid states are updating based on velocity. And as they're updating, also these cues are coming in simultaneously into the sensory area.
I'm not talking about novel environment. I'm talking about the top one.
Oh, the top one over here. And this is in familiar environments, right? So
if there is another if instead of the bird, you have something squirrel.
Yes, then it will reconstruct the grid state associated previously with the squirrel.
But there is no location information, consistent location information.
We have fed it. So by calling it a familiar environment, I should have clarified, I'm sorry. So in a familiar environment, what that means is that the agent is moved through the environment with, through its self-movement. So it's integrating its velocity inputs, grid states are being generated, and then visual inputs are, landmarks are being seen, and those are being associated through this associative learning rule here, right, on the sensory. That's what it means to have a familiar environment. You've moved through the environment and gone through and done association of the seen inputs with the internal velocity updated grid states. And then you associate them, and after associating them, then that counts as a familiar environment. And then in that environment, you could, if now I show you a picture of one of the landmarks, you will reconstruct where you are and vice versa. Okay, so this model, because of the very large space of the grid codes, is capable of doing mapping, basically, of a large number of environments with pretty small catastrophic forgetting, not the memory cliff, but like the more the catastrophic forgetting. So this is just a paradigm from the Moser lab where animals put into environment 1 and then tested again in environment 1, then put into environment 2, tested in 2, and so on out to 11 environments. But then after seeing 1, it doesn't see 1 again, and it just kind of learns them sequentially one after the other. And then at test time, then after that, then all the environments are tested sequentially, and then they're tested again with no sensory cues. And the takeaway over here is that what's happening is that in vector hash model, the after learning, even up to 8 or 9 environments, and there's landmarks at every environment. So learning means the association between grid states and the landmarks. The memory is the agent is able to remember the correct associations out to like many different rooms. And by contrast, if this is a model where, again, the grid and hippocampal states are permuted relative to each other, and then bidirectional learning takes place in them, then this is the performance of that model. Yeah. Oh, yeah.
Learning multiple environments, and you have to sort of navigate the space of grid codes. Do you initialize like environment 1 is here, and then you have to like allocate enough grid code? So environment 2 is here, or they connect?
That's a good question.
How are you thinking about
Yes.
where things go in that space of
Yeah.
connector, don't connector, overwrite, or do?
So in this experiment, it was just completely random. Where each time, exactly, each environment just complete random. We did not look to see overlap, but because the representational space is so big, you know, random choices end up being extremely non-overlapping in this case. So.
But random choice there essentially means randomizing the phase.
That's right. Random, not the phase relationships within a module, but across modules. That's right. Across modules. That's right. You just pick a random phase for each module, and that's it. So statistically, of course, there could be a collision, which is like, you have to be careful about that. Yeah. So, okay. So so intermediate summary over here is that, you know, you can do, you know, it's possible, you know, to have one-shot learning of the cues and environment, you know, cues in an environment with internal estimates of position in the environment. You can do, you know, inference on novel paths, and you can get a good capacity without catastrophic forgetting with this model. Okay, and now to the, you know, sort of most interesting, in a sense, property of this network is the following, which is that now let's go back again to only thinking about the scaffold, and the claim is that this model not only generates a large scaffold of individual fixed points, but it's capable of storing a very large scaffold of a large sequence scaffold. So it can memorize or have a very long sequence in the network. And how does this work? So you know, we create, you know, a continuous sequence of arbitrary shape, and now okay, by hand. And then we ask the model to learn this sequence, the scaffold to learn it in the following way, which is that the hippocampus has to construct a two-dimensional velocity vector, which corresponds to the next step, right? The next, you know, which would move the grid state to the next position in this design sequence. Okay, and so we can construct a sequence and successfully, with a small MLP that goes from hippocampus into 2D, we can now memorize this circuit. This grid circuit can memorize something like 14,000 step sequence with like just a few hundred grid cells and hippocampal cells. And we can also do like a hairpin curve, a Hilbert curve, and other curves over here. So by contrast, if you look at the same size hippocampal network and then use like directional associative learning in a Hopfield network, the same size network can recover only about 51 steps of this sequence. And if we add in the benefit of you know, this error correction from the scaffold, it can go as up to 102 steps. And then we can also consider that the way the sequence is learned is that you have a state in hippocampus, which then projects up to grid cells, and then you have a learned projection. I'm sorry, and then you have a projection. You have grid cells represent a state. They project to hippocampus, which then learns to project into grid cells to the next state in the sequence. Okay, and this also does very poorly. So it's interesting, this model does much better than this model at generating sequences. And then again, we can build a long episodic memory, like actual memory, by now heteroassociating sensory inputs onto that scaffold, the sequence scaffold. And, you know, I'm not going to belabor this, but basically we can get the same kinds of memory curves for sequence memory. Okay, and again, we can compare with other memory models. So here's vector hash, number of trained transitions versus number of actually correctly retrieved transitions, and you can see that this is basically retrieves everything that it's trained on, whereas, you know, these other models fail all the way down here. Yeah.
Well, don't worry about
Yeah.
time, at least.
Oh, you do?
No, you're getting worried, but no, yeah.
Oh, okay. Oh, okay.
We're
Okay.
for an hour and a half.
Oh, right. Okay, great.
But, two back, why did you feel it was important to train the velocity vector off the campus? I mean, because one could have done it in the grid space, which is kind of very natural. You can take a step in a grid space, fully deterministic, what that step would be and so forth.
So the grid cells are only grid cells, so we don't have any extra cells that can learn the transition structure. But I think we could do it within entorhinal cortex. So you're saying, why does this arrow into the velocity input come from hippocampus? Why doesn't it come from the sensory layer? And the answer is it could, but with a caveat. So remember that the sensory patterns that are recalled are not the exact same pattern that was stored. Right? And so, and in fact, the quality of that sensory recalled pattern is degrading as you have more and more patterns stored. So that's a dynamical quantity. So it's not as good. But what you could do is you could learn the velocity transitions off of the hippocampal reconstruction of the sensory states. Because what's interesting is that the hippocampal reconstruction of the sensory state is the same always. So that's reliably the same, no matter how noisy the sensory the state is. So we could have indeed, instead learned it from some cortical state, or
I think I'm asking kind of a
Yeah.
basic question. So there's a little MLP there,
Yes.
right?
Yes.
You could have put that little MLP from grid to grid.
I could have put the MLP from grid to grid.
Lost grid, right? Because the grid is a unique place in space, so then there's
Yeah.
that you want.
That's a good question. So, the grid states are very correlated. They have a lot of structure because, as you're advancing in grid phases within a module, right? There's a lot of, the same a given module goes through the same state transitions, a lot. Even, it'll do that here in space, and then it'll do that here in space, because this is repeating every lambda, right? Each module. It's only collectively that they're kind of spanning this very big space. And the question is, are those states different enough that you can robustly learn a good velocity vector? And the answer is, we haven't tried that, but I'm going to write that down. We'll try that too. Yeah, so another good suggestion. Yeah.
So the role of hippocampus is what? Is random expansion?
Essentially, it's random expansion in this model. That's right. But with the random projection. Yeah. So it's related to some of the things that, you had thought about and written about on olfactory system. Okay, so that's what this does. And let me see. Okay, and there's weak dependence on number of MLP units. It's just linear. So now the conceptual question: why is this working? Okay, so why does this model do what it does, and why do other models not do what big good sequence memory? So the idea is, if you're going through some episode over here, in this case Goldilocks and the Three Bears, if in the traditional Hopfield network with asymmetric connections, if you're at Goldilocks and then you want to recall that the next item is the cabin of the bears, the network has to both encode in the information for Goldilocks and what she looks like and so on, and then it also has to have that state, has to have enough information to know exactly which neurons to activate for the next state, which is again a very rich state, which is the house, right? And so there's a lot of information that needs to be passed from the Goldilocks state to the house state, which is to reconstruct that whole state. Okay, so there's a high dimensional transition vector that has to go from Goldilocks to the house. And which way do you go? Okay, by contrast, in this model, all that needs to happen is, for the sequence part, you only need to remember a 2D vector, right? Which direction and distance do I go in 2D? Okay, so it's a much lower, for any given state, you only need to know within 2D where to go next. Okay, and so this is sort of this idea. So you're learning just these 2D transition vectors, and then all the actual content is done by this heteroassociation. Yeah.
Simpler conceptually implementation of that, you'll have shift register. You have a chain of neurons.
Yes.
Coding for position in one dimension.
Yes.
And then you just shift register.
Pretty much. Pretty much. That's exactly right. You don't even need the fancy stuff. Yeah, I mean, this only just gives you more, you can just have all of these different sequences, like if it matters, because with the shift register, if you only have 1D chain of neurons, shift register, you only have one such sequence. But with this 2D sheet, suddenly you can do many, many sequences, many different ones. Each one is very long. So of course, even in the 1D case, you could just say one episodic memory is this chunk of that 1D chain. This is this chunk. For 2D buys you a lot more, right, than 1D. So it's basically that. It's just that in 2D. Exactly. Okay, and just to show you that this isn't just, this was our hypothesis that this is the reason why it works. And we can kind of like test this sort of quantitatively by saying, how about, we can, within the grid space, okay, so the question is, what is the set of possible velocities that are allowed, right? So we're saying that the input velocity has to shift you to the next state. But you could say, well, the states have to be contiguous within some distance of each other, right? So that means that, you have a direction and then you have distances that are only within some ball. So that's less information. If the ball is bigger, you need more information because you have more different possible vector lengths, right? So we can just vary that ball that's allowed for the next state transitions. And what we find is the number of correct transitions drops with the number of allowed next state distances. And in fact, it drops exactly like you would expect, which is like the log of the number of states that are possible for the next step. Okay, so in other words, it is really about like the amount of information that needs to be recovered to specify the next step. All right, fine. So the interim summary here is that, we can construct now a scaffold, but for sequences instead of fixed points, and that we can build long sequences with it. Okay, so that's sort of the idea of the episodic memory model. We've got item memory, we've got spatial memory. And now the question is, what about there's such a rich phenomenology of findings and experiments in hippocampus. To what extent is this model consistent with that phenomenology? And so there's a whole bunch of beautiful experiments that people have done. And I guess without describing in great detail, I'll just say that it's possible by using the vector hash model together with the idea of just remapping to random locations in the representational space. We can generate, the same model generates splitter cells, it generates route-dependent cells, it generates directional tuning cells, task-dependent cells. And it's, we can try to understand, we can use the model to try to understand what could be possible mechanisms for memory consolidation, hippocampal memory consolidation. So in this model, what we can consider is that we have a bunch of different inputs here. This is like the Goldilocks house and an armchair and a bed. And some of these items are presented just once, okay? And so these heteroassociative weights are learned, once. And then some of the items are presented multiple times. And each time that item is repeated, you just make an additional update to that heteroassociative with the associative learning rule. Okay, and so then, if we don't consolidate if we just kind of run through all the patterns once, okay, then if we consider the effects of lesioning the hippocampus, so if you lesion hippocampus and you look at the sensory recovery error, okay, so in the case where each pattern is just seen once, then this is kind of the pattern of errors that you get as a function of percentage of cells lesioned. Very quickly, you have a lot of error in your reconstruction. But now if we have some patterns that are unrepeated and some patterns that are repeated, then the repeated patterns after lesion are much more resistant to the lesion, and the unrepeated patterns are much more prone to forgetting after the lesion. So that seems to work. And an alternative version of consolidation could have been that each time you see the same input pattern, you bring a new scaffold state into play and associate it to that second. Like I see the same item twice, and I associate it with the new scaffold, and then a new scaffold point, and a new scaffold point. That doesn't work. So the prediction here would be that the consolidation process is not associating the same input with multiple fixed points, but it's just building a stronger association with the same fixed point. So I mean, it's a hypothesis, but it remains to be tested. Yeah.
Is there biological entorhinal lesion or silencing results that support your theory and that there's maybe not other non-grid mechanisms involved?
I mean, I think you mean in the context of just general episodic memory, it's very difficult because for spatial navigation experiments, yeah, I mean, I think that's a good question. So oh yeah, I also for time I took these out. So yes, so there's some very interesting results on lesion, and the lesions experiments were sort of very confusing to people thinking about spatial representation in the circuit, because the question was like, between grid cells and place cells are both playing a role in spatial representation, which one is really doing the spatial, which one's driving that spatial tuning first? Okay, and so the studies were that if you lesion grid cells and then test the animals in a familiar environment with lots of cues, place cells have nice tuning. Their tuning properties are intact. But if you lesion place cells, then the grid cell tuning curves degenerate. They get less sharp. So the takeaway from those findings, experimental findings, was people thought maybe that means that place cells are fundamental, right? Because they're necessary for grid cell tuning. And on the other hand, but grid cells are not necessary for place cell tuning. And so the thought was, then there's these, for example, these continuous attractor grid cell models can't be right, because these models are saying grid cells are internally generated, they're fundamental, and place cells are the things that remap and are not so fundamental, right? And so how do you reconcile those? And so what's interesting here is that, so in this model, you can do those lesions one by one, and if there's any noise in the velocity input, the model predicts that so if you lesion place cells and there's noise in the velocity inputs to grid cells, the grid cell tuning will degenerate because grid cells are deriving their tuning by velocity integration, but the velocity is noisy, so they're not well tuned, and the sensory inputs come in to correct the state via hippocampus. So because hippocampus is lesioned, those states cannot be corrected, and the grid tuning should be poor. But the prediction is that the cell-cell relationships among grid cells should still be there. And in fact, data analysis of the lesion studies subsequently showed that, in fact, the cell-cell relationships are preserved even when hippocampus is lesioned. Okay, and conversely, the prediction would be that if animals after grid cell lesion are tested in an environment with no external cues, right? Like a familiar environment, then you put the animal in, you turn out the lights, and then let the animal explore. The prediction of this model would be you will not get hippocampal tuning, and that experiment has not been done. So, but that's that's a prediction.
But what is your prediction about lesioning respect to episodic memory? If you lesion grids?
If you lesion grid cells and what happens to episodic memory, the prediction would be that there would be a pronounced episodic memory deficit. Yeah. Okay, so that brings me to like this question of method of loci. And actually, this Saturday, there is the World Memory Championships taking place at MIT, I think at Kresge Hall. And like it's an annual event. Like how many of you have heard of memory championships? Oh, okay, some of you heard of it. Okay, so people who went to medical school have definitely heard of it. So this method of loci is something that memory athletes use. And so this is a method that was really old. Like people like Cicero have talked about it, in writings. And so the idea, what is the method? So okay, so where is it used? Okay, so there are people who are memory grandmasters, kind of like chess grandmasters, and they can do things like these crazy feats, like memorize 1,000 random digits in 60 minutes, memorize the order of 10 decks of cards in 60 minutes, memorize the order of one card deck in less than 2 minutes, like just randomly shuffled deck, right? And this guy, Clemens Meyer, used a 300-point long journey through his house to memorize 1,040 random digits in 30 minutes. Yeah, and it's used a lot, and it's super reliable, and it's not an inborn talent. It's a skill that can be cultivated. So like in a 10-minute talk, someone can like walk you through the methodology, and you can be memorizing like 30-word sequences. Like it's pretty impressive. Okay, so what is the technique? The technique is that you imagine a place that's a very familiar place to you, and you imagine taking a path through that space, this route. And so you go through, for example, your TV, a couch, of your childhood home, and then as you're going through the deck of cards, what you do is you just imagine, the first card, you go to your bedside table and you put the card on that table, and then you go to your pillow and then you place the next card on your pillow, and so on. And you associate these cards with the locations in this route. And then at the time for a recall, you just kind of walk through your house and just pick the cards back up again, and that's how you remember, okay? And it's very counterintuitive to me because, to remember a list of items, you're now adding to your memory burden because you have to now remember which item goes where, right? So like, why does this work? Why does that make it better, right? That seems very strange. Okay, and as far as I could tell, there was no such circuit model. Okay, and so, but then, thinking about like these scaffolds in vector hash made us realize that maybe there's a way to understand why this technique works. It's shockingly effective, and how it could work. Like, how do we model this? Okay, so how do we leverage, previous, right? What they're doing is they're leveraging previous sensory learning, right? Because they've, they have this experience, and they have this memory of it, and they're leveraging it in some way to do this new learning. Okay, so you could say, fine, maybe what's going on is somehow you've got this prior memory, right? In vector hash, this is all our, our space over here. And then we get some new inputs, right? Like some new sensory data, like this mnemonic input that we need to memorize. And we just extend the model by just associating these mnemonic inputs to the sensory states and reconstructing them. But the problem with that is that, we had this like memory continuum, right? And if already in your episodic memory, you're in your continuum regime, you're not having high fidelity recall of these items because these items are not being remembered with high fidelity, right? And I'm sorry. Okay, I take it back. I'm sorry. I should have. Okay, I misspoke. Okay, let's just think about why don't we just use vector hash as vector hash to store these new items, right? And the critique there is we can't just directly associate these mnemonic inputs onto hippocampus like vector hash, because then we're in the continuum regime and we don't get accurate recall, right? And so, if we're out here, we're not going to accurately recall these new states. So there's no point, okay? So, but there's one really interesting fact about even when you're in this continuum regime, which is that even though recall of the sensory state is not accurate in the continuum regime, it's faithful, it's reliable in that whatever corrupted state is recalled, it's every time, if you cue it with a different version of the noisy input or a different version of the noisy input, you always recall the same slightly corrupted pattern. Okay, so by that I mean, if this is the true sensory input or this is the true sensory input, if this is the recall of vector hash, and after 2000 states, this might be what this recalled pattern looks like, and this may be what this recall pattern looks like, even if you cue it with the non-noisy state, or if you cue it with some noisy version of this pattern, it'll always retrieve this exact pattern. Okay, even though this pattern is not the same as the sensory pattern, but it'll be reliable. So it's not accurate, but it's reliable. Okay, so that means that the recalled sensory states are reliable, okay, even if they're not the same as what was stored. So then we can now think about, right? So this is a diagram showing that, basically what happens is if I start out with two inputs to the circuit, maybe one input is here and one input is here, and the green dots here are the actual, the true sensory states that were the patterns that were put into the network. Then this network restores, like it reconstructs a state, which is not necessarily exactly that dot, but every time it recalls that state, it's the same state. Okay, even if the input pattern was noisy in different ways. I hope that makes sense. I think I'm seeing some confused faces. But okay, so the fact that it's reliable means that then we can use the mnemonic inputs as long as we associate them with the recall sensory state, not with the true sensory state that was used in learning, but with the recall, the reconstruction of the sensory state, that becomes part of the scaffold. Okay, so what I'm saying here is now we're going to associate this mnemonic input to this recollected sensory state over here. And then this memory palace now has these items in this items here have, now we can think of this whole network as a scaffold. We can think of these as abstract, reliable states. And now these inputs can be associated to them. Okay, and so before the bottleneck was like hippocampal size, now the bottleneck is the size of the sensory, area, which could be all of, a lot of neocortex. And so basically, even though these sensory states might, be degraded versions of the true sensory data, right? But the new items that are associated onto those corrupted sensory states are now reliably recovered. Okay, so, and reliably and faithfully recovered. And so the idea here is just that you've extended the scaffold with these recall sensory states, and now you can kind of attach new inputs to those states. Okay, so basically the other thing that you can do too is you can flush the palace if it's possible to renew these connections. So what memory athletes do is that they might use the memory palace for today's competition, but then if they want to use a memory palace for tomorrow's competition, they won't use the same palace. They have a set of palaces, and they'll use some other ones, and then it takes a few days, and then that palace will be available for use again. So I don't know exactly how they actively forget. It's a very interesting question for us, I think, as neuroscientists, but that's what they do. Yeah, Sam.
Okay, so as far as I know, the method of loci wouldn't work with just using an empty room for the memory palace, right? It seems like familiar sensory information is important. But I was just thinking, like on the logic of the scaffold, right? As long as I can locate myself on the scaffold and use get the vector. So imagine I said, like, imagine an empty room, start in the corner, move 1 foot forward, in a particular direction, right? And then remember, that card there, right?
That's not good enough because you really need to, you need the sensory, because the sensory states, like your bottleneck here is the dimension of your sensory recalled state. And so, the richer that sensory recall, the better. It's a better scaffold. It's just a bigger scaffold. It's just that your knee of your memory curve is at the size of that. At the size of that, because that's how many synapses you have, right? Like, just think about that as your, the number of plastic synapses is the number, the number of associations you can build between whatever your internal recalled state is and that external cue. And the bigger that is, the better your ability to remember.
Let's imagine some arbitrarily rich sensory representation of the items you're trying to memorize.
Yes.
But I thought, maybe I just misunderstood that. I thought that as long as you can locate yourself in the grid, then you can heteroassociate that content with that point in the grid, right? So just wondering why can't this? Is it like path integration? You get cumulative noise?
No, it's not that. Yeah. It's that, because even here, right? Like, even in vector hash original, the sensory inputs couldn't be directly associated with the grid. They needed to be associated with this dimensionally expanded random projection from the grid states, because that sets the capacity of the memory network. So it's about that. It's a capacity is set, the knee of this memory curve, the point at which you enter the memory continuum, that is set by the dimensionality of the sort of the bottommost point of the scaffold that you associate to. So that's kind of the idea. Yeah. And so here, both because, you're free to flush, right? Like, if, you don't want to flush your whole episodic memory, right? Like, because you don't want to forget everything that you've learned so far about all your interactions you've had with people in order to remember that deck of cards. You want to keep that. And there you might be deep in the continuum, but you want to have like fresh memory for like, your memory tournaments. And so you want to just build these new associations onto those and flush them. Okay, and in fact, that is the point. In fact, one of the predictions here should be that, one of the predictions is that you don't necessarily need spatial memory, right? You should just be able to use episodes that may not be even spatial, but you should be able to use those for memory palaces. They don't have to be physical palaces. right? That would be a prediction. Another prediction would be that, the states that you access during memory palace recall should be more correlated with like where you are in that palace, like in an abstract sense, like the grid states should be reliably, reproducible at each of those locations in the palace, more than the actual sensory data that you first that evoked the neural responses when you first built that palace, right? So because the sensory cues that you're recalling, when you're recalling your palace, the states may be quite different from the states that were evoked by the sensory data that built that palace, right? Because you're not recalling, you might be deep in the memory continuum. So you're not really remembering the items faithfully, but it's so that would be the prediction. Okay, I had one more part, but I'm looking at the time, and even though Bernardo says it's okay, I don't want to, right, Bernardo? I should.
We started we're an hour and a half there.
Yeah, okay. So I think that's it. So I won't go to the last part, but I'm happy to talk about it later about like, mapping other structured metric domains with the same network, but happy to do that post talk. So okay, so just to summarize, basically, in fact, I'm not gonna, you can read these. I don't want to read through these. I just wanted to make a few points. So we should think about this model of hippocampus as a memory network, where hippocampus is a pointer to content, but it doesn't contain any content. And that's the contrast with the autoencoder network. So the autoencoder network is kind of like JPEG. It's saying, I get data, the data is compressible, I compress the data through the bottleneck, and then I store it in this compressed form. Here, this is saying there's not a compression, hippocampus is not a compression engine, it's a pointer. That's why we call it hash, because it's like a hash code. It's just like content independent pointer to like items out there in sensory cortex for reconstruction, right? That is the primary difference. The things that enable the network are like this very structured grid states that are low dimensional. That allows you to do like high dimensional sequence memory. And there's a ton of work to do in the future, like thinking about much more realistic models of hippocampus. We don't have many different hippocampal fields. We only had one hippocampus. Of course, I think some of us know that, some of you guys know better than I do, all the richness in hippocampus. And so there's a lot to do there. Hierarchical memory is something that we haven't touched, memory consolidation, abstract reasoning, and also, relating this to, entorhinal hippocampal connectomes, which, by the way, are coming down the pipe in a newly funded project with David Tank, Sebastian Sung, Viren Jin, and Honghui Zhang. So I look forward to that. Yeah, thank you very much.
This is so cool and beautiful. Thank you so much. So I have many questions, but I'm picking my favorite. So my question is, could the vector hash model speak to kind of in memory interferences or confabulations in kind of, I know you mentioned that the space was so massive that the likelihood of selecting the same exact state phase module combination is very unlikely, but like in the actual, in a rodent or in a human, is that space equally as massive, or do you think that those kind of phenomena are more as a result of like a consolidation, reconsolidation, retrieval of that phase?
That is a very good question. And in fact, actually, your question just gets to one of the things that this model just doesn't do. So I think, this notion of hierarchical memory. So here right now, for example, if we just take item memory, like the first part without any sequence or episodic nature, each memory input is like a completely independent item. And because it's a pointer out to content without any content, there is no sort of similarity structure baked in any way. And so there's no way for this model to store information based on similarity of the content. It really treats each input as completely independent. Now, in the episodic memory case, it is the case that items nearby in time are actually placed, have similar coding because, we're advancing the grid states, in a continuous way. And so it's kind of like there's a temporal contiguity similarity sort of thing in there, but that's the only notion of it. It's not content. And so I'm very interested in the question of like how you can build, content-based, associations in memory. Open question.
Yes, thank you, Dr. for that.